Hello everyone, welcome to today's CellTalk. Today our presenter is Ms. Soumyadatta Basak. She will be presenting on the topic gene therapy mediated partial reprogramming extends lifespan and reverses age-related changes in aged mice. Before going to the presentation, let me first introduce Soumyadatta. Soumyadatta Basak works at Renix Life Sciences as junior bioinformatician. Primary responsibility involves identifying and implementing bioinformatics pipelines tailored to project specifications. She is currently working in a project focused on neoantigen peptides, employing molecular docking techniques to investigate their binding interactions with specific receptor proteins for potential applications in cancer immunotherapy. She holds a master's degree in bioinformatics from Chandigarh University, and before that, she earned a BSc in genetics from Guru Nanak Institute of Pharmaceutical Science and Technology. Soumyadatta has gained valuable experience through dissertation, training, and internships at institutions such as National Institute of Animal Biotechnology, Hyderabad, and Bionome. Her goal is to continually acquire new skills and apply them to impactful projects within the realm of bioinformatics. So I welcome Soumyadatta to present the research paper that she has selected.
Thank you for the nice introduction. So I'm going to present the research paper, which is basically the gene therapy mediated partial reprogramming extends lifespan and reverses age-related changes in aged mice. The original author of this paper is Makib, and this paper was selected from a journal called Cell Reprogram, which has an impact factor of 1.2, and it was published in the year of 2024. And this research has an impact on lifespan and health span. So let's get started with the introduction. So as we know that aging is the primary risk factor for most of the diseases, be it cardiovascular diseases or neurological disorder or even cancer. And there has been researches on regenerative medicine that has been going on recently, and it has highlighted or shown its potential uh, to reverse the age by targeting age-related cellular changes. So the particular approach that this study is using is called partial cellular rejuvenation by using the factors called Yamanaka factors. So these Yamanaka factors includes these four factors or proteins, you can say, which is OCT4, SOX2, KLF4, and CMYC. And these together has shown potential in reversing age-related epigenetic changes, offering a promising approach to age reversal. So here is the general background. It's important to know what is epigenetic information loss, which is basically gradual changes in gene expression pattern over time leading to aging and tissue degeneration by disrupting cellular function. So these Yamanaka factors that we discussed earlier, this has been discovered by a scientist called Yamanaka in the year of 2012. So he found out that these factors has a potential to act like stem cells, and if not controlled properly, it might lead to cancer, and especially this CMYC factor, which is present in the OSK factor. So, in this study, we are using partial cellular reprogramming instead of the full one. So, in this process, we are excluding the CMYC factor, and we're only taking this three factor, which is OCT4, SOX2, and KLF4 factors, Yamanaka factors, and we are going to see how these factors affects the age reversing process, and this is done without fully reprogramming the cell into stem cells, which might lead to tumor. And next we have the present challenges. So translating the whole process of partial reprogramming into animal models and then from animal model to human trials is quite challenging because of its differences between genetic complexity, lifespan, and there are also potential safety reasons. For example, we just discussed it has the risk of tumor formation, and it the release of these factors into the body needs to be done in a very controlled manner, and it's very tricky to do so. And if not done in a controlled manner, it might lead to serious side effects. And then we have the objective of the research. The objective of the research is to test whether the systemic delivery of OSK genes by a vector, which is adeno-associated virus vector, can extend the lifespan and improve health in extremely old mice, translating to potential age reversal therapies for humans. So here's the methodology. The first step of this methodology section is generating the AAV vectors. So two viral vectors have been used in the study. In the first vector, these Yamanaka factors has been incorporated and been injected into the mice. And in the second vectors, a compound known as doxycycline has been injected and then given to the mouse, which this doxycycline helps to control these three factors, which are Yamanaka factors. So after injecting, this is the second step, which is mouse studies and frailty scores. So the population has been divided into two groups. One is the control group and one is the experimental group. In the control group, they are treating the mouse with doxycycline, but not with the OSK treatment. And along with that, they're also giving them a phosphate buffer solution, which is basically harmless, and these things does not affect lifespan or health span at all. While on the other hand, the experimental group of mice is receiving the OSK treatment and also the doxycycline at 1 week interval. And these has been after injecting, they have been studied. The researchers tracked the physical health of mice by using a thing called frailty index, which basically measures reflexes like motor skills, body condition, sensitivities, and this helped them to determine if the treatment was actually improving the mice health or not. The next step is to measure the age-related DNA changes. So what it's been done here, so after injecting the tissue from liver and heart is taken, and they have been taken to test the DNA methylation patterns. So first they took the control group and they took the tissue from the control group and they tested the DNA methylation test, and after that they test the same thing with the experimental group. And DNA methylation is a key marker of aging, and they use this to check if their treatment reserved aging in these tissues or not. And this will help to compare between the control group and the experimental group and how the OSK treatment is actually affecting them. So the next step, we have RNA extraction and testing. So in this particular step, the tissue has been taken from the mice, and then we extract the RNA. The scientists extract the RNA, and the RNA is converted into cDNA by a technique called qPCR. And it is a process that has been done to measure how active certain genes are in the mice. This helped the scientists to understand how the treatment affected the genes involved in aging. So this is basically the step where they are doing the epigenetic testing process. Next, this is the second part of the methodology section, in which the whole process, which is the partial reprogramming, is being tested on human scale cells or keratinocytes. But in this case, they are using lentivirus to deliver the OSK treatment instead of AAV vector. And after injecting with the OSK factors, the cells have been infected with a chemical called pyronomine in spheres. It's a chemical that is used to kill to kill the cells, but not the virus. So after 23 days, the cells which has survived the whole process, the treatment, they were taken and they were studied to see how the aging markers had changed and their protein were analyzed. So the last step is the immunoblot analysis. So to check if the proteins which are like OCT4, SOX2, and KLF4 are actually being made in the experimented mice or not, immunoblot analysis were performed. So the proteins ran through a gel and which basically separates them by size. It transferred to a membrane, and then special antibiotics were added to them to detect the presence of the proteins, the specific proteins. And these proteins were then visualized by a chemical reaction that produces light, allowing them to see which proteins were actually present in the cells. Next, we come to the result. So the first thing we can see here is the median lifespan extension. So the mice who received the OSK treatment, the median lifespan of them is around 142.5 weeks. While when they compared the control group, the median lifespan extension is only 133 weeks. So here we can definitely see an improvement how the median lifespan extension is happening from 133 to 142, and it definitely suggests that uh, it increases the lifespan of the mice. Not only the lifespan, when they tested the frailty scores, they found out that the treated mice also exhibited improved health matrices, suggesting that OSK not only extend the lifespan, but it has the potential to enhance the overall health during aging. Next, we have the reduced epigenetic age in liver and heart. As we discussed, that the tissues have been taken from liver and heart and been tested for any epigenetic changes or not. So this is what they found. So OSK expression led to the significant decrease of epigenetic age of liver tissue and as well as heart tissue, which definitely indicates the genomic methylation pattern has been restored in the liver function. And this the overall results have implication rejuvenation implication. These findings highlight that the potential of OSK mediated partial reprogramming to rejuvenate key organs, supporting the notion that cellular rejuvenation can positively impact health during aging. So this is the results. So in the first part of the figure, we can see that the vectors have been injected to 124 week old mice, and one is the control, and the other one is the OSK treated approach. So after that, when they are doing the analysis to find out the median lifespan, so they see that the OSK treated mice had 142 around 140 to 143 weeks of median lifespan, while the control group which did not receive any kind of OSK treatment, their median lifespan was only about 130 weeks. And when they further analyzed the remaining lifespan of the control and the experimented mice groups, they found out that even in the remaining one, the OSK treated mice had a much longer lifespan compared to the controlled mice group. And and the last section of this figure, we can see the frailty index graph. So in the control mice, we can see the frailty increase index is quite higher, which suggests that they kind of have like motor skill issue and aging issue. And in the OSK treated OSK treated mice, we can see there's a very less frailty index, which suggests that the overall treatment definitely enhance the lifespan and the health span. So this is the second part where they are testing the epigenetic age of heart and liver. So we can see here the control and the OSK treated mice. The epigenetic age of the OSK treated mice is definitely lesser than the epigenetic age of the control mice, as we can see here in the graph. And lastly, when this is basically where they are testing the human skin cells. So these two are the control. We can see the epigenetic age of there are almost about 100, but when they are treating it with OSK in different concentrations, the epigenetic age is definitely decreasing here to like almost 80 years. So this is the overall results of the human skin cells. So 65-year-old male patient, the the human skin cells were taken from the 65-year-old male patients, and they were treated with the OSK treatment. Treated keratinocytes showed significant epigenetic age reversal compared to control groups, and the results indicate that the potential of Yamanaka factors to rejuvenate human somatic cells efficiently. Next, we have the discussion section. It is divided into six parts. Actually, we are going to see one by one. So first is lifespan extension. We definitely saw in the earlier slides that the median lifespan of the experimented mice group, which received the OSK treatment, is 142 weeks, and the one who did not receive any treatment, their median lifespan is 133 weeks. So we can see a definitely an improvement in this part. And then we have improved health outcomes. So we also saw the frailty scores graph, which definitely increased that the OSK treated mice had definitely better lifespan and health span compared to the control one. And lastly, potential for human therapy. The whole thing was tested on human skin cells as well, and findings suggest that the partial rejuvenation could be promising therapeutic avenue for extending lifespan and health span and lifespan in humans also. So there is limitations in the studies. So this study only focused on male mice. So there might be potential differences between male and female responses, and female responses need to be studied for this particular approach. And next we have future research direction. So optimizing AAV vectors. So future studies should aim to improve how we can improve the systemic delivery of the vectors in a more efficient way so that the scientists can control the overall factors, OSK factors. Otherwise, it might lead to tumor, as we discussed earlier. And tissue specific analysis. Further research is needed to evaluate how partial reprogramming affects various tissues, but not only liver and heart tissues, but tissues from all of the body at a molecular level. So this is the conclusion. First one we can see that revolutionary potential. This study shows promising result or promising potential for partial reprogramming as the transformative approach in regenerative medicine and anti-aging therapeutics. Lifespan extension and improved health outcomes in aged mice demonstrate that epigenetic rejuvenation could slow or reverse aging. Next, we have human studies. Definitely, this study was tested on human skin cells, but it would be more encouraging if we see more studies on humans are going on this particular uh, therapy. And a thorough investigation into systemic delivery methods and long-term effects will be crucial for future clinical implications. And here we have the impact of the research. So first one is the breakthrough in age reversal. So this study definitely demonstrated that the partial cellular reprogramming by using the Yamanaka factors can definitely reverse the epigenetic aging and could improve the lifespan and health span, potentially leading to novel anti-aging therapies. As we can see the comparison between the OSK treated mice and the ones who were not treated with the OSK factors. And then this study also provides a critical foundation for developing safe and effective rejuvenation therapies for human, which can be beneficial for future studies. If you have any further questions, you can find me at my account.
Thank you, Somadatta, for that interesting research paper presentation. If audience has any questions, the forum is open for questions. You can write the questions in the chat box. I don't see any questions. So with this, we conclude our today's CellTalk presentation. The next presentation details will be conveyed to you by email. Thank you, everyone.